In this tutorial, we will see how to predict backbone chemical shifts of a protein by back calculating them from the PDB structure solved by nuclear magnetic resonance spectroscopy or NMR, and separately from a structural ensemble that is generated by the web server SHIFTX2, the tutorial for which you have seen already. The web server that we will be using for prediction or back calculation of the chemical shift is SHIFTX2, which is this one right here. And after prediction of the backbone chemical shift values, that is for the carbon, nitrogen, oxygen, and hydrogens in the backbone of the protein, we will compare our values and benchmark them against the experimental backbone chemical shift values deposited in the BMRB database or the Biological Magnetic Resonance Data Bank, which is this web server out here. So SHIFTX2 can make predictions of both backbone and side chain 1H, 13C, and 15N chemical shift of proteins, which are these isotopes of the specific elements here, hydrogen, carbon, and nitrogen. And by utilizing an advanced machine learning technique which incorporates combination of sequence-based and structure-based predictions of chemical shifts. So it makes use of a combination of support vector regression, support vector machines, and artificial neural networks to precisely train a set of 197 proteins and further test them on a separate set of 61 proteins. So as you can see, it is a very extensive and robust prediction method, and more often than not, it is highly accurate. Now, a problem that you might face in this regard while predicting the chemical shift values is the degeneracy of the particular chemical shift, especially for that matter in case of the hydrogen chemical shift. You might see that these values are actually coinciding or correlating with each other quite often for different systems and different proteins. So in that case, a better assessment would be via the C alpha chemical shift or the C beta chemical shift, or from a secondary chemical shift, which is the difference between the actual chemical shifts of C alpha and C beta predicted by SHIFTX2 and the random coil chemical shift that are predicted by using different algorithms. So just to give you an example, if we go to this website proteinnmr.org, it'll give you an idea about the random coil chemical shifts which are predicted by different methods and assessed by different groups. And so the difference of the actual chemical shift and this random coil chemical shifts will give you an idea about the secondary structure propensities of the protein in that particular residue region or domain of the protein. So if you go to the secondary structure features, and you can see the secondary chemical shift is predicted as the difference between the delta value that is observed, which is in parts per million, and the delta of the random coil, which is also in parts per million. The C alpha atoms in alpha helices, for example, will tend to have a positive secondary chemical shift, and C alpha atoms in beta strands will tend to have a negative chemical shift. Whereas the C beta atoms behave exactly opposite to that of C alpha atoms, and they will have a positive secondary chemical shift in case of beta strands, whereas a negative chemical shift in case of the alpha helices. So you can see the secondary chemical shift can predict the propensity of different regions to sample different secondary structures accordingly, and this is a very useful metric to assess the quality of the predicted chemical shift in case of a degeneracy or significant overlap between different systems and the experimental values out there. Okay, so we will back calculate these chemical shifts first from a static structure that is solved by NMR spectroscopy, that is a PDB structure, and then from an ensemble of structure generated from that static structure and sampled via the SHIFTX2 web server for running a molecular dynamics simulation in the coarse-grained method. Okay, so in order to first back calculate the chemical shift from a static structure, that is a PDB structure, we need a PDB structure in the first place. So the model system in our case is a solution NMR structure of a beta hairpin of alpha-synuclein, intrinsically disordered protein that's responsible for the pathogenesis of Parkinson's disease. It's a neurodegenerative disorder. And the PDB code for this is 4BXL, So this is a structure of alpha-synuclein in complex with an engineered binding protein, which is also known as an affibody. So affibodies are like antibodies, but are small and robust proteins engineered to capture or bind a large number of target proteins or peptides with high affinity. Hence the name affibody. So in this case, it captures the toxic beta hairpin of alpha-synuclein. So the beta hairpin conformation of alpha-synuclein from a predominantly disordered ensemble or a disordered state of the IDP, alpha-synuclein. This particular beta hairpin conformation, which is the beta sheet in the monomeric form, is responsible for initiating the self-assembly and aggregation of the alpha-synuclein monomeric peptide. The beta hairpin is found in the residue range from 35 to 56 in this PDB file, and there are 10 different conformers that are submitted here, and as with all NMR structure, these are represented in the ensemble form out here. So we will have to select one particular structure, and that will be annotated as the most representative structure in this ensemble of structures. And we will use that structure as an input to the SHIFTX2 web server and generate the chemical shift by back calculating them. So this is the PDB code of the structure, that's 4BXL, and this is the publication that led to that particular beta hairpin structure. It's called Sequestration of a Beta Hairpin for Control of Alpha-Synuclein Aggregation, and is by the Hoyer group, who have been a pioneer working in this neurodegenerative disorder field for a long time now. Okay, so let's first go ahead and download the structure in the PDB format. So you go to download files and then PDB format, and then save it in your specific workspace directory, which is workspace 3 for me. So 4BXL in the PDB format saved into the workspace 3 directory. So once you save it, it automatically opens for me in the Chimera software package. So I'm just going to click on it, and you'll see the structure of the 10 conformers solved by NMR spectroscopy, the solution NMR. And this is the beta hairpin structure having the 10 conformations that's bound to the affibody protein, also having the 10 conformations. So if we hover over the residues, we'll be able to see the respective chain IDs that the beta hairpin and the affibodies correspond to. So this guy out here is for the affibody and it belongs to chain B. The beta hairpin here belongs to chain C, and the another portion of the affibody, another fragment of the affibody, belongs to the chain A. So we'll just need chain C for this analysis by the SHIFTX2 chemical shift predictor, and also just one conformation, which is the most representative conformation of the beta hairpin out here. So if we close this and just open the PDB file in the text format, so that's 4bxl.pdb that we have just downloaded, you'll find that there's 10 different models in this, and the most representative structure will be annotated out here with the remark 210, which is the best representative conformer in this ensemble is 5. So that's model number 5 in this PDB file. So if we go to the different models out here, so we will find, first of all, there's model 1, and then there's model 2, 3, 4, 5, 6, 7, 8, 9, and 10. Okay, so we'll get rid of all of these different models from 1 to 4 and from 6 to 10, and just retain model 5, as well as we'll also have the chain C only of model 5 and get rid of chains A and B. So that is only the beta hairpin structure for the alpha-synuclein. So let's close this and go back to Chimera, because we'll be performing the rest of the operations in Chimera out here, and just open the structure again in Chimera. So if we do that and we go to favorites and then the model panel, you'll be able to see all the different models out here. So we can view all of these models as a list here itself by clicking on group or ungroup, and once you do that, you'll find all the different models that are being listed out here. So in this case, if you just select model 1, then model 2, model 3, so you can do a separate selection for each of these conformations that are submitted in the PDB file out here. Okay, so The chain, again, that we are concerned about is chain number C, and the best representative conformer here is model number 5. So we'll go ahead and use the command line interface in Chimera in order to do the selections. So first of all, let's say we select model 5 out here. So the command for that is select #0, which is the PDB file as a whole in the form of the model number, and then .5, which is the submodel, and which is also the separate fifth conformation inside that PDB file. So 0.5 annotates that particular representative conformation of the beta hairpin structure along with the affibodies, that is all the three chains together. So once we press that, it does that particular selection as is shown in red out here on top of the already present other models. Now if we go to select and then invert all models, which means it will do an invert selection on model number 5, that is, it will select everything except model number 5. So if we do that invert selection, then it selects model number 1 to 4 and 6 to 10, but not model number 5. And then if we go to actions and then atoms and bonds and then delete, it will delete the atoms or bonds of all the other models apart from model number 5. So aside model number 5, it deletes the atoms or bonds of all the other different models from 1 to 4 and 6 to 10. Now we need to do a particular selection of the chain IDs A and B out here, because chain ID C is the beta hairpin which we are concerned about, and we'll save that as a separate PDB tab. So in order to do the selection of chains A and B, we'll type into the command line select #0.5 colon all.A or #0.5 colon all.B. Okay, and then press enter, and you'll find that the selection is highlighted here, which in this case, in my case, it's in red actually. You'll probably find it in the color of green fluorescence for you, okay, in Chimera. All right, once you do that selection, and you can see that chain number C is not being selected, which means these are the chains that we are going to delete, the atoms or the bonds of these chains. So go back to actions again, and then select atoms or bonds, and then delete. So you see just the beta hairpin of the best representative conformation from the PDB, that's conformation number 5, is retained, and that corresponds to residues from 35 to 56. So this is the N terminal and that is the C terminal. So we'll go ahead and save this file as a separate PDB. So save PDB, we go to the workspace directory first and then give it a name. Let's say as underscore hairpin.pdb. Okay, and we use untransformed coordinates again. So save this one, and we then close this and go back to the SHIFTX2 web server out here. Now SHIFTX2 has this several input options. So the first one is the protein deuterated, meaning is the hydrogen atoms in the protein are replaced by the deuterium heavy atom, that is an isotope of hydrogen, which is no in our case because we have only hydrogen atoms in the PDB file. And then the pH for generation of the chemical shifts or the prediction of the chemical shifts. So we'll use a physiological pH, that is pH 7, which is the neutral pH in which all biological processes occur inside the body. And then the temperature, which is already set to 298 in Kelvin. So that's the optimum temperature inside the body in the physiological conditions. And then you have an option for calculating the phosphorylated residues, which is basically a post-translational modification. So we don't have any phosphorylated residues out here. So we'll keep that option as no. And then select the type of chemical shift to be predicted. So here you have options for all shifts, backbone chemical shifts, and side chain chemical shifts only. So we'll go ahead and select backbone chemical shifts only because we are not concerned about the side chain chemical shifts. And then you have the output format, which could be in a tabular format or the CSV comma separated format, or the NMR star format, which is a specific format used by the BMRB or Biological Magnetic Resonance Data Bank, which we'll see in a minute. And then you have the NEF format. So we'll keep it in the tabulated format, which is easier to follow. and then combine results with shift y, which is another function, and is very efficient, and it precisely correlates with that of the experimental chemical shift values. So certain regression models will be generated in order to correlate the chemical shifts values by using this shift y function out here. And then you have the option for the PDB ID. So you can input directly the PDB ID out here, which in our case is 4BXL, but then again 4BXL has this 10 different conformations or models together. So we are not going to go ahead with this option for the moment, and we are just going to deal with the static PDB structure that we just constructed first of the beta hairpin, and then we predict the chemical shift from the ensemble of structures that is generated by the SHIFTX2 web server. Okay, so we are going to upload a local PDB file, which is choose file, and then we go to the workspace directory, which is this one out here, and then we select the as_hairpin.pdb that we just generated. So just select that and open. So it uploads that file, and then multiple chain options is basically if you have more than one chain, but in our case it's just a single chain. That's chain number C for the beta hairpin. So that's it, and you're all set, and go ahead and submit. Once you do that, once you press on submit, it will take a while to give you the results in the next page, and immediately you can see the results are predicted in the next page with the backbone atoms. And in the first column you have the residue numbers, and then you have the residue names in the one letter code. So starting from the glutamic acid and glycine and valine, then you have the C alpha chemical shift values, you have the C beta chemical shift values, you have the carbonyl carbon chemical shift values, you have the nitrogen chemical shift values, the hydrogen, and the alpha hydrogen out here. Okay, and these values for the C beta, which are shown in star, is not determinable, probably because of the position they are in inside the protein structure. That is, they might be sterically hindered or restricted in order for them to be, predicted properly, which you can also find from the experimental chemical shift values, that they won't be properly annotated as well, probably, and we are expecting that. Okay, so once you have these values, you're going to go ahead and download the prediction. So we first right click on this file location and then save link as, and it will ask you to save the file in a specific extension with the CS format out here. So the file naming would be .cs, which is the short form for the chemical shift. So I'm just going to rename this file so that it's easier to recognize, because we'll be doing the chemical shift analysis for the ensemble of structures as well. So I'm going to just rename it as static. So as_hairpin_static recognizes it as the static PDB file from which the chemical shifts were predicted, and just save it in the workspace directory here. So once we go ahead and hit save, so you can find this structure will be able to open in the text format itself. So before that, before going through this file, I'm just going to go ahead and go back to the chemical shift prediction web server, SHIFTX2, and keeping again all the other options as before and some of the options as default, I'll just go ahead and load up the previously generated multi-model PDB file by SHIFTX2 web server. So I already have generated the ensemble of the structures of the beta hairpin PDB by using the SHIFTX2 web server, which is saved here as multi-model.pdb, which has the 10 different models in the form of an ensemble. So model 1 to model 10 out there. So just going to load this up and then hit submit again. And in the next page, you will see the results in the form of an average predicted chemical shift of all the 10 different models. So this guy out here is not just of a single static PDB file, but of all the 10 different models, an average value of that. And we'll compare this value with the values that are predicted for the static PDB file. So just go ahead and download the predictions again. Save link as, let's say, multi-model. We name it as _ensemble.cs, and then save this out here. And we'll peruse this later on, but before that we also need the experimental values in order to compare with the predicted chemical shift values of both the static model and the average chemical shift values that are predicted for the ensemble of the beta hairpin structure. So I'm going to go ahead and go to this BMRB database. So a little bit about the BMRB data bank, which is basically user submitted, or the experimentalists doing NMR experiments, they submit the data for the NMR observables out here, which may include chemical shift, the J coupling constant, the paramagnetic resonance enhancement, and sometimes these are combined with the small angle X-ray scattering, or SAXS data as well, which is a different repository that you can get the data from. But here, this is only for the nuclear magnetic resonance observables that you get from the NMR spectroscopy experiments. So you have a few fields to search for the specific protein of your interest if an experimental structure for that is available. If the experimental structure is not available and is not solved by NMR, then you won't find that data out here. And also subject to the fact that the experimentalist actually chose to submit the data out here. Sometimes they do not, like they want to keep it discreet and confidential. So that's why you might not find a data submitted for a particular PDB structure that exists in the protein data bank. Okay, so you have a few fields here. First is the entry ID, which is the specific accession number. So if you know the accession number of that particular PDB in this BMRP database, then you can go ahead and type in that accession number. Then you have the PDB ID itself. It is this option that we'll pursue. So we'll type in the PDB ID. You have the title as well, so the title of the protein, which in our case is alpha-synuclein. We could use that as well, but then again you would end up with a number of different solved structures for alpha-synuclein and the corresponding chemical shift data to it. Then you have the author name, that is the publication which gave rise to that particular structure, and any of the author names in that particular publication. And then you have the name for the molecule. So there's a whole bunch of options which you can combine together in order to search for your specific protein for which a NMR structure is available and solved, and all the NMR observables should be available there in that structure. Okay, so in our case, since we already know the PDB ID, we are just going to go ahead and type in 4BXL out here. Okay, and hit search. And once you do that, a new tab will open and it will try to look for your particular search, and there you go. You have it here with the entry 19353, and it has this chemical shift validation, which is based on certain chemical shift assignment. The data that we are interested in is the NMR star file, which is an interactive viewer, which is available in the form of an interactive viewer as well as an NMR star text file. Okay, so if you scroll down further, just before downloading the data, you'll find that the 13C chemical shifts with a count of 450. So there are a total of 450 carbon chemical shifts here. And the nitrogen chemical shifts have a number of 113, and the hydrogen chemical shifts have a number of 744. So, and these are the assigned chemical shift lists that have been assigned previously before performing the NMR spectroscopy. Okay, so if you go down further, you'll find the entity assembly ID, which means there are two chains for the Affibody out here, for which the chemical shifts are also predicted, and they belong to the IDs 1 and 2, and then ID 3 belongs to the alpha-synuclein 35 to 56 residues forming the beta hairpin. So that has this entity ID of 2, but the assembly ID is 3 out here. Okay, and these two together belong to the entity ID of 1. And then you have the specific amino acid residues for the entity IDs, for the corresponding entity IDs. So this is belonging to entity assembly IDs 1 and 2, whereas entity ID 1, and this is only for the Affibody, and these are the residues that belongs to that particular Affibody. And then you have the 35 to 56 residues belonging to that of alpha-synuclein, which is in total 22 residues. And you have the corresponding weights as well given in Daltons. Okay, so another information and an important one as a cross-reference is basically the related database links. So in this case, PDB, it's very pertinent information in case you're looking for other alpha-synuclein structures, maybe not a beta hairpin, but still belonging to the same protein class. And so you have 4BXL, which is this one out here, when you have 1xQ8, which is basically a lipid-bound alpha-synuclein structure, which you saw in the other tutorial that we had for the molecular modeling. Then you have 2KKW, which is also the PDB structure of alpha-synuclein solved in a membrane-bound environment. Then you have 3Q27. So these are all the alpha-synuclein structures that are available in the protein data bank and corresponds to that of the 4BXL structure, which is a fragment of the full-length alpha-synuclein out here. Okay, and you have the reference database for the NCBI as well for the protein sequences that you can find for the corresponding alpha-synuclein peptide. So we're going to go ahead and have a look into the NMR star structure here, which is in the text format. So just click on that. It can show you the text formatted file in the browser itself. So you see all this information, data, entry information, and then the category and the frame code and everything else. So the entry type is that of macromolecule, and the date of submission and the date of accession is given here, which is 2013, which probably corresponds with the date of the publication that gave rise to the structure. And the method of experimental determination is NMR, and the type of solvent used is solution. So there could be solid state NMR determination of structures as well without the use of a solvent. Okay, then you have the description of the protein here. So an engineered binding protein to alpha-synuclein was selected from a combinatorial protein. So the main information that we are looking for amongst the others that is available out here are the chemical shifts themselves. So if you scroll down further, and we scroll down to the very end of the file here, where you have the assigned chemical shift allotted. So this is assigned chemical shift list, and then if you go further down, you will find the assignment of the chemical shifts with the respective corresponding assembly ID and the entity ID and everything. So let's go through the columns here. So the first column, the very first column, is for the chemical shift ID, which is basically the sequential atomic chemical shift that are being solved here. So this includes all the atoms, including the hydrogen, carbon, and nitrogen atoms together. And the next thing that you have, the next column that you have, is the assembly atom ID, which is this guy out here, and it has not been allotted an assembly atom ID. And then you have the entity assembly ID that you saw earlier, which is basically 1 for the Affibody. And then you have the entity ID, which is also 1 for the Affibody and 2 for the beta hairpin structure. Then you have the complex index ID, that's 2 here, and then you have the sequence ID, which is the sequence of the residues that are being generated, that the chemical shifts are being generated for. So that's 2 out here, starting from 2 and then 3 and then 4. This is for the Affibody. And then you have a whole bunch of other informations, including the residues and the specific atom types and the specific atoms here that are being predicted for. And then you have the residue IDs, or the residue numbers, for which the chemical shift is predicted. So you will find that for the carbon atom, the chemical shift is quite high, whereas for the hydrogens, the chemical shifts are quite low predicted here. So this is the column for the chemical shifts. So 8.107, 4.030, although this is beginning with the Affibody itself. So if you want to go down and get to the part where the beta hairpin chemical shift assignment starts, so if you go down further, and we are basically looking for number 2 on this column, which is the entity ID that we are looking for the beta hairpin. So if you scroll down, so this is where the number 2 starts from. So this is belonging to the beta hairpin, and this is just an average of the ensemble of the structures that the NMR PDB had. So this is what we are concerned about. So we'll go ahead and copy this, but before that, you might notice that the sequence IDs or the residue IDs for the respective for the beta hairpin is starting from 1 here, as opposed to 35 in case of the PDB. This is because the assignment itself records the very first residue there. Even though it's 35 in the PDB, it will record it as number 1. So we'll have to change these residue IDs in order to compare directly with the predicted values, or the other way around, that we change the residue ID of the predicted chemical shift file that we have there. Okay, so just select this part and copy this further down and up until here, and then we copy this and paste it in a text file which we will name as experimental hairpin underscore experimental hairpin dot cs. Okay, and then we paste this here. Now we'll have to extract two particular columns from here. This applies to that of the predicted chemical shift files that we have there for both the static structure and the ensemble. And we will organize this in such a manner all the C alpha assignments will be put in one file, and the C assignments that is belonging to the carbonyl carbon will be put in another file, whereas the H assignments would be put in a separate file, and the N assignments will also be put in a separate file. So we'll do the same for the predicted chemical shift values in the files that have been written already before. So just going to save this one first, and let's have a look at the predicted chemical shift file for the static structure first, which is this one. If you open that, you'll find we'll have to save this column and all the rest corresponding columns belonging to the separate, predicted chemical shift values for the atoms here. So just for the sake of this tutorial, we'll just focus on the C alpha atoms, let's say. Okay, so we pick column 1 and column 3. We'll write small awk scripts which you're familiar with already because we have dealt with it in the previous molecular modeling tutorial that we had, if you had a chance to go through it. And so the awk is a very powerful scripting language that could be used to process files, especially text files, easily without having to manually intervene and everything. So just close this, and so we know already that this is column 1 and column 3, and we'll have to skip the first two lines here in order for it to recognize it as column 1 and column 3. So, and we'll renumber these residues as well, starting from 1 until that of 22. So 35 to 56 is 22 residues, so that it corresponds well with the assigned chemical shift experimental values that we have just downloaded there. Okay, so the script that we will write here is awk, and then we skip the first two lines. So nr is number of records greater than 2, and print the first column. So if the number of records are greater than 2, that is exceeding the second line, we'll print the first column and the third column, which is the C alpha predicted chemical shift. Okay, and then we name the input file here as hairpin static.cs, and there you go. You have the two columns that you were looking for. So the first one is the residue ID, and the second one is basically the chemical shift values that you have. Okay, so now we need to write this into a new file so that it's easier to access by the XMGrace program that we'll use to plot these chemical shift values and compare them and benchmark them with that of the experimental values. So before that, as I said, we need to rearrange the residue numbers, which we can also do by including an additional command in this awk script. So in order to start from residue number 1 here, so nr greater than 2, and then print $1, which stands for the first column, and we'll subtract 34 for each of those numbers there. So if you subtract 34 from 35, it will lead to 1, 34 from 36 will lead to 2, 34 from 37 will lead to 3, and so on and so forth, and we'll ultimately end up with total number of residues from 1 to 22 out here. So comma, and again $3, and then the name of the input file, which is as_hairpin static, and then save it in a new file. So let's say ca static.cs. So this stands for C alpha of the static conformation, C alpha predictions of the static conformation in the chemical shift extension format. Okay, so save this. If we open this new file, you'll find that all these fields are written properly. Okay, so these additional numbers that you have here is because of the blank spaces at the end, which we can get rid of by selecting them and deleting them. So that's fine. Okay, so 1 to 22 here, that's all done. And then we escape this and save this file separately and have a quick look at the predicted chemical shift values for the ensemble. So that's again multimodal ensemble.cs. You'll find the same format here. So column 1 corresponding to residue numbers and column 3 corresponding to C alpha values. So we'll go ahead and save these column 1 and column 3 and readjusting the residue numbers of column 3. Type in the same command, just gonna copy this out here, and now the input file would be multimodal ensemble.cs and write it in a new file, ca ensemble.cs, and save it, and you'll find that the C alpha predicted chemical shifts are written corresponding to their respective IDs that are numbered from 1 to 22. Once again, delete these last two lines out here real quick and then save this file. Okay, so that's done. We have two columns, with the first column being the residue numbers and the second column being the C alpha predicted chemical shift values for both the static structure and the average values from the ensemble structure. Now we need to do the same for the NMR assigned chemical shift experimental values that we have just downloaded there in a text file. So the name of that file is exphairpin.cs, and if we look through this file, we'll find that column number 1 is corresponding to that of the specific ID of the BMRB that the assignment was made for, so we are not concerned about that. Column number 2 is empty. Column number 3 corresponds to that of the assembly ID. Column number 4 corresponds to that of the entity ID, which is 2 in our case. And then you have column number 5 and 6, which are the residue IDs respectively. But then again, if you want to make a selection for only the C alpha atoms from here, let's say, so we need to pick this 1, 2, 3, 4, 5, 6, 7, 8th column as well, and annotate those as C alpha atoms, and their corresponding residue IDs will also get picked in that manner. So we need to do a selection for the C alpha atoms based on 1, 2, 3, 4, 5, 6, 7, 8. So the 8th column out here. So we can achieve that by writing a small awk script again. So awk $8 here. So $8 corresponds to that of the 1, 2, 3, 4, 5, 6, 7, 8. So these are the atomic IDs that you have, and we'll pick specifically C alpha. So if $8 is equal to CA, then write column number 1, 2, 3, 4, 5, 6 that corresponds to the residue ID, column number 6, and its corresponding chemical shift, which corresponds to 7, 8, 9, 10, 11. So 11th column there. So write column number 6 and column number 11 if column number 8 is equal to CA. Simple enough logic. So column number 6 and column number 11. So that's it, and we have the name of the input file here, which is exphairpin.cs. If you press enter, you'll find the corresponding residue IDs in the first column and the chemical shift values for only the C alpha atoms in the second column being printed on screen. So we can go ahead and save this in a separate file. Let's name it as CAEXP. So the C alpha chemical shift predictions for the experimental values .cs. Okay, we'll use a program called XMGrace. So XMGrace is a 2D plotting tool that allows to interactively modify plots and save figures in the format of your choice. So it's a very efficient plotting tool, plotting program. But you don't need to use this strictly. So you can also use Excel if you want to make your plots. So the way XMGrace works is on the files directly. So it makes a choice according to the columns. So it regards the first column as the x-axis and all the rest of the columns as the y-axis. So if you have more than one plot, then it can regard all the rest of the columns apart from the first column as the y-axis and make the plot accordingly. So it's efficient in that regard, but you can always resort to other plotting programs if you want. So I'm just going to write down the names of the file followed by the xmgrace command itself. So that's going to be caexp.cs. That's the experimentally assigned chemical shift values for the C alpha atoms, followed by the predicted chemical shift values for the static structure, the PDB structure of the beta hairpin, followed by the predicted chemical shift values of the average chemical shift values of the ensemble that we predicted via the SHIFTX2 web server. So if you press enter, then it will make a plot immediately of the three values that you provided for the chemical shift, the three columns taken from the three different files. And the first line is the experimental chemical shift value here, which is in black. So this guy in black is basically the experimental chemical shift, and you find that for residues 35 to 56, which is now changed to 1 to 22 out here, but you can clearly see that for the beta hairpin, the predicted chemical shift values match up very well with that of the experimental chemical shift values. So you can go to the set appearance and then change the width of the lines that it generates. So the first line, that is for the experimentally assigned chemical shift values, is in black, and so we can type in the legend as experiments, and the second one is in red, which is for the static structure predicted chemical shift values. So we can type in the name as static out here, and the third one is the average values that are predicted from the ensemble. So you can type in ensemble here. Okay, so you can just change the color to a more prominent green and then apply, and you'll see the three plots being plotted together. So except for residues in the towards the end of the C terminal, that is in here, there's a little bit of deflection with the predicted chemical shift values, that is for both the values, for both the static and that of the ensemble predicted chemical shift values. These are of slightly higher values. So for the last two residues, that is 21 and 22, let's say. But apart from that, rest of the predicted plots pretty much match up with that of the experimental values. So that's great. So if we click on the axis to change the labels there, so we can have for the y-axis chemical shift, and the unit should be in parts per million, that's ppm. Apply, and then you go to the x-axis and name it as the residue index, and then apply as well. Okay, so we can save this file in a image format. That's, you go to print setup and then you select, let's say, a PNG image. So give it a name, let's say chemical shifts .png, and apply, and then print it there itself. So once you do that, you'll find that the file has been printed and the image has been generated for the corresponding chemical shifts. And as you can see from the plot, the predicted chemical shift for both the static structure and the ensemble, they correlate and correspond well with that of the experiments. So you can employ other statistical methods like regression analysis and correlation coefficients in order to find the degree of fit of the predicted chemical shift values with that of the experimentally assigned chemical shift values. Okay, so SHIFTX2 is a very efficient program in that regard to predict the chemical shift from a PDB structure, in this case the beta hairpin of alpha-synuclein bound to a Affibody.